Genetic purging. Genetic purging is the increased pressure of natural selection against deleterious alleles prompted by inbreeding. 1. Purging occurs because deleterious alleles tend to be recessive, which means that they only express all their harmful effects when they are present in the two copies of the individual, i.e., in homozygosis. During inbreeding, as related individuals mate, they produce offspring that are more likely to be homozygous so that deleterious alleles express all their harmful effects more often, making individuals less fit. Those less fit individuals pass fewer copies of their genes to future generations. In other terms, natural selection purges recessive deleterious alleles in inbred individuals. Purging reduces both the overall number of recessive deleterious alleles and the decline of mean fitness caused by inbreeding, the inbreeding depression for fitness. The term, purge, is sometimes used for selection against deleterious alleles in a general way. It would avoid ambiguity to use purifying selection in the general context, and to reserve purging to its more strict meaning defined above. The mechanism. Deleterious alleles segregating in populations of diploid organisms have a remarkable trend to be, at least, partially recessive. This means that, when they occur in homozygosis double copies, they reduce fitness by more than twice than when they occur in heterozygosis single copy. In other words, part of their potential deleterious effect is hidden in heterozygosis but expressed in homozygosis, so that selection is more efficient against them when they occur in homozygosis. Since inbreeding increases the probability of being homozygous, it increases the fraction of the potential deleterious effect that is expressed and, therefore, exposed to selection. This causes some increase in the selective pressure against partially recessive deleterious alleles, which is known as purging. Of course, it also causes some reduction in fitness, which is known as inbreeding depression. Purging can reduce the average frequency of deleterious alleles across the genome below the value expected in a non-inbred population during long periods. 2. Which reduces the negative impact of inbreeding on fitness. If inbreeding is due just to random mating in a finite population, due to purging the mean fitness declines less than would be expected just from inbreeding and, after some initial decline, it can even rebound up to almost its value before inbreeding. Another consequence is the reduction of the so-called inbreeding load. This means that, after purging, further inbreeding is expected to be less harmful. The efficiency of purging is reduced by genetic drift and, therefore, in the long term, purging is less efficient in smaller populations. 1. Purging can be increased if individuals mate with relatives more often than expected by random mating. The joint effect of inbreeding and purging on fitness. Accounting for purging when predicting inbreeding depression is important in evolutionary genetics, because the fitness decline caused by inbreeding can be determinant in the evolution of diploidy, sexual reproduction and other main biological features. It is also important in animal breeding and, of course, in conservation genetics, because inbreeding depression may be a relevant factor determining the extinction risk of endangered populations, and because conservation programs can allow some breeding handling in order to control inbreeding. 3. In brief, due to purging, inbreeding depression does not depend on the standard measure of inbreeding, writes inbreeding coefficient F, since this measure only applies to neutral alleles. Instead, fitness decline it depends on the purged inbreeding coefficient, G, which takes into account how deleterious alleles are being purged. Purging reduces inbreeding depression in two ways. First, it slows its progress. Second, it reduces the overall inbreeding depression expected in the long term. The slower the progress of inbreeding, the more efficient is purging. A more detailed explanation. In the absence of natural selection, mean fitness would be expected to decline exponentially as inbreeding increases, where inbreeding is measured using Wright's inbreeding coefficient F. 4. The reason why decline is exponential on F instead of linear is just that fitness is usually considered a multiplicative trait. The rate at which fitness declines as F increases, the inbreeding depression rate delta, depends on the frequencies and deleterious effects of the alleles present in the population before inbreeding. The above coefficient F is the standard measure of inbreeding, and gives the probability that, at any given neutral locus, an individual has inherited two copies of a same gene of a common ancestor, i.e. the probability of being homozygous, by descent. In simple conditions, F can be easily computed in terms of population size or of genealogical information. F is often denoted using lowercase f but should not be confused with the coancestry coefficient. However, the above prediction for the fitness decline rarely applies, since it was derived assuming no selection, and fitness is precisely the target trait of natural selection. Thus, Wright's inbreeding coefficient F for neutral loci does not apply to deleterious alleles, unless inbreeding increases so fast that the change in gene frequency is governed just by random sampling, i.e., by genetic drift. Therefore, according to the model, the decline of fitness can be predicted using, instead of the standard inbreeding coefficient F, a purged inbreeding coefficient, G, that gives the probability of being homozygous by descent for partially recessive deleterious alleles, taking into account how their frequency is reduced by purging. 1. Due to purging, fitness declines at the same rate delta than in the absence of selection, but as a function of G instead of F. This purged inbreeding coefficient G can also be computed, to a good approximation, using simple expressions in terms of the population size or of the genealogy of individuals, see box 1. However, this requires some information on the magnitude of the deleterious effects that are hidden in the heterozygous condition but become expressed in homozygosis. The larger this magnitude, denoted purging coefficient D, the more efficient is purging. An interesting property of purging is that, during inbreeding, while F increases approaching a final value F equals 1, G can approach a much smaller final value. Hence, it is not just that purging slows the fitness decline, but also that it reduces the overall fitness loss produced by inbreeding in the long term. This is illustrated below for the extreme case of inbreeding depression caused by recessive lethals, which are alleles that cause death before reproduction but only when they occur in homozygosis. Purging is less effective against mildly deleterious alleles than against lethal ones, but, in general, the slower is the increase of inbreeding F, the smaller becomes the final value of the purged inbreeding coefficient G and, therefore, the final reduction in fitness. This implies that, if inbreeding progresses slowly enough, no relevant inbreeding depression is expected in the long term. Implies, for example, that the average fitness of a population that has been moderately small for a long time can be very similar to that of a large population with more genetic diversity. In conservation genetics, it would be very useful to ascertain the maximum rate of increase of inbreeding that allows for such efficient purging. Predictive model for the joint effects of inbreeding and purging caused by a reduction in population numbers. Consider a large non-inbred population with mean fitness W. Then, the size of the population reduces to a new smaller value n. In fact, the effective population size should be used here, leading to a progressive increase of inbreeding. Then inbreeding depression occurs at a rate delta, due to partially recessive deleterious alleles that were present at low frequencies at different loci. This means that, in the absence of selection, the expected value for mean fitness after T generations of inbreeding would be. Whereas the population mean for Wright's inbreeding coefficient after T generations of inbreeding. 4. However, since selection operates upon fitness, mean fitness should be predicted taking into account both inbreeding and purging, as in the above equation, is the average, purged inbreeding coefficient, after T generations of inbreeding. 1. It depends upon the purging coefficient, D, which represents the deleterious effects that are hidden in heterozygosis but exposed in homozygosis. The average, purged inbreeding coefficient, can be approximated using the recurrent expression. There are also predictive equations to be used with genealogical information. The example of inbreeding depression due to recessive lethals. Average population fitness W against number of generations T of maintenance with population size n equals 10 when, in the original population, each gamete carried on the average one rare recessive lethal. Evolution of W expected only from inbreeding, red line, or from inbreeding and purging, blue line. As an example of genetic purging, consider a large population where there are recessive lethal alleles segregating at very low frequency in many loci, so that each gamete carries on the average one of these alleles. Although about 63% of the gametes carry at least one of these lethal alleles, almost no individual carry two copies of the same lethal. Therefore, since lethals are considered completely recessive, i.e., they are harmless in heterozygosis, they cause almost no deaths. Now assume that population size reduces to a small value, say n equals 10, and remains that small for many generations. As inbreeding increases, the probability of being homozygous for one or more of these lethal alleles also increases, causing fitness to decline. However, as those lethals begin to occur in homozygosis, natural selection begins purging them. The figure to the right gives the expected decline of fitness against the number of generations, taking into account just the increase in inbreeding F, red line, or both inbreeding and purging, blue line, computed using the purged inbreeding coefficient G. This example shows that purging can be very efficient in preventing inbreeding depression. However, for non-lethal deleterious alleles, the efficiency of purging would be smaller, and it can require larger populations to overcome genetic drift. Relevance to the evolution of reproductive systems. Inbreeding depression and purging play a major role in the evolution of reproductive systems. As an example, they determine when selfing becomes at an advantage compared to outcrossing. 5-6. Another example is the genomic renewal in yeasts. Saccharomyces cerevisiae and Saccharomyces paradoxus have a life cycle that alternates between long periods of asexual reproduction as a diploid, ending in meiosis that is usually immediately followed selfing, with only rare outcrossing. 7. Recessive deleterious mutations accumulate during the diploid expansion phase, and are purged during selfing. This purging has been termed genome renewal. 8-9. Evidence and problems. When a previously stable population undergoes inbreeding, if nothing else changes, natural selection should consist mainly of purging. The joint consequences of inbreeding and purging on fitness vary depending on many factors: the previous history of the population, the rate of increase of inbreeding, the harshness of the environment or of the competitive conditions, etc. The effects of purging were first noted by Darwin 10 in plants, and have been detected in laboratory experiments in invertebrate populations undergoing inbreeding in zoos or in the wild, as well as in humans. 11. The detection of purging is often obscured by many factors, but there is consistent evidence that, in agreement with the predictions explained above, slow inbreeding results in more efficient purging, so that a given inbreeding F leads to less threat to population viability if it has been produced more slowly. 12-13. Nevertheless, in practical situations, the genetic change in fitness also depends on many other factors, besides inbreeding and purging. For example, adaptation to changing environmental conditions often causes relevant genetic changes during inbreeding. Furthermore, if inbreeding is due to a reduction in population size, selection against new deleterious mutations can become less efficient, and this can induce additional fitness decline in the medium-long term. In addition, part of the inbreeding depression could be not due to deleterious alleles, but to an intrinsic advantage of being heterozygous compared to being homozygous for any available allele, which is known as overdominance. Inbreeding depression caused by overdominance cannot be purged, but seems to be a minor cause of overall inbreeding depression, although its actual importance is still a matter of debate. 14. Therefore, predicting the actual evolution of fitness during inbreeding is highly elusive. However, the component of fitness decline expected from inbreeding and purging on deleterious alleles could be predicted using G. Understanding genetic purging and predicting its consequences is of great importance in evolutionary and conservation genetics. Endangered populations used to undergo inbreeding due to their reduced numbers, and purging can play a relevant role in determining their extinction risk and the success of conservation strategies. 15. References. DNA profiling. DNA profiling, also called DNA fingerprinting and genetic fingerprinting, is the process of determining an individual's deoxyribonucleic acid (DNA) characteristics. DNA analysis intended to identify a species, rather than an individual, is called DNA barcoding. The DNA double helix. DNA profiling is a forensic technique in criminal investigations, comparing criminal suspects' profiles to DNA evidence so as to assess the likelihood of their involvement in the crime. 1. 2. It is also used in paternity testing. 3. To establish immigration eligibility. 4. And in genealogical and medical research. DNA profiling has also been used in the study of animal and plant populations in the fields of zoology, botany, and agriculture. 5. Background. Sir Alec Jeffreys, pioneer of DNA profiling. His discovery led to the conviction of Colin Pitchfork in 1988. 1. Starting in the mid-1970s, scientific advances allowed the use of DNA as a material for the identification of an individual. The first patent covering the direct use of DNA variation for forensics, US 5593832A, S5593832.PDF, 6-7, was filed by Jeffrey Glassberg in 1983, based upon work he had done while at Rockefeller University in the United States in 1981. British geneticist Sir Alec Jeffreys independently developed a process for DNA profiling in 1984 while working in the Department of Genetics at the University of Leicester. Jeffreys discovered that a DNA examiner could establish patterns in unknown DNA. These patterns were a part of inherited traits that could be used to advance the field of relationship analysis. These discoveries led to the first use of DNA profiling in a criminal case. 8, 9, 10, 11. The process, developed by Jeffreys in conjunction with Peter Gill and Dave Orett of the Forensic Science Service (FSS), was first used forensically in the solving of the murder of two teenagers who had been raped and murdered in Narborough, Leicestershire in 1983 and 1986. In the murder inquiry, led by Detective David Baker, the DNA contained within blood samples obtained voluntarily from around 5,000 local men who willingly assisted Leicestershire Constabulary with the investigation, resulted in the exoneration of Richard Buckland, an initial suspect who had confessed to one of the crimes, and the subsequent conviction of Colin Pitchfork on January 2, 1988. Pitchfork, a local bakery employee, had coerced his co-worker Ian Kelly to stand in for him when providing a blood sample. Kelly then used a forged passport to impersonate Pitchfork. Another co-worker reported the deception to the police. Pitchfork was arrested, and his blood was sent to Jeffreys' lab for processing and profile development. Pitchfork's profile matched that of DNA left by the murderer, which confirmed Pitchfork's presence at both crime scenes. He pleaded guilty to both murders. 12. After some years, a chemical company named Imperial Chemical Industries, ICI, introduced the first ever commercially available kit to the world. Despite being a relatively recent field, it had a significant global influence on both criminal justice system and society. 13. Variations of VNTR allele lengths in 6 individuals. Although 99.9% of human DNA sequences are the same in every person, enough of the DNA is different that it is possible to distinguish one individual from another, unless they are monozygotic identical twins. 14. DNA profiling uses repetitive sequences that are highly variable. 14. Called variable number tandem repeats, VNTRs, in particular short tandem repeats, STRs, also known as microsatellites, and minisatellites. VNTR loci are similar between closely related individuals, but are so variable that unrelated individuals are unlikely to have the same VNTRs. Before VNTRs and STRs, people like Jeffreys used a process called restriction fragment length polymorphism, RFLP. This process regularly used large portions of DNA to analyze the differences between two DNA samples. RFLP was among the first technologies used in DNA profiling and analysis. However, as technology has evolved, new technologies, like STR, emerged and took the place of older technology like RFLP. 15. The admissibility of DNA evidence in courts was disputed in the United States in the 1980s and 1990s, but has since become more universally accepted due to improved techniques. 16. Profiling processes. DNA extraction. When a sample such as blood or saliva is obtained, the DNA is only a small part of what is present in the sample. Before the DNA can be analyzed, it must be extracted from the cells and purified. There are many ways this can be accomplished, but all methods follow the same basic procedure. The cell and nuclear membranes need to be broken up to allow the DNA to be free in solution. Once the DNA is free, it can be separated from all other cellular components. After the DNA has been separated in solution, the remaining cellular debris can then be removed from the solution and discarded, leaving only DNA. The most common methods of DNA extraction include organic extraction, also called phenylchloroform extraction, 17, Chelix extraction, and solid phase extraction. Differential extraction is a modified version of extraction in which DNA from two different types of cells can be separated from each other before being purified from the solution. Each method of extraction works well in the laboratory, but analysts typically select their preferred method based on factors such as the cost, the time involved, the quantity of DNA yielded, and the quality of DNA yielded. 18. RFLP analysis. Restriction fragment length polymorphism. RFLP stands for restriction fragment length polymorphism and, in terms of DNA analysis, describes a DNA testing method which utilizes restriction enzymes to cut the DNA at short and specific sequences throughout the sample. To start off processing in the laboratory, the sample has to first go through an extraction protocol, which may vary depending on the sample type or laboratory SOPs, standard operating procedures. Once the DNA has been extracted from the cells within the sample and separated away from extraneous cellular materials and any nucleuses that would degrade the DNA, the sample restriction fragment length polymorphism. RFL. Each method of extraction works well in the laboratory, but analysts typically select their preferred method. The cell and nuclear membranes need to be broken up to allow the DNA to be free in solution. Once the DNA is free, it can be separated from all other cellular components. The cell and nuclear membranes need to be broken up to allow the DNA to be free in solution. Once the DNA is free, it can be separated from all other cellular components. After the DNA has been separated in solution, the remaining cellular debris can then be removed from the solution and discarded, leaving only DNA. The most common methods of DNA extraction include organic extraction, also called phenylchloroform extraction, 17-Chelix extraction, and solid-phase extraction. Differential extraction is a modified version of extraction in which DNA from two different types of cells can be separated from each other before being purified from the solution. Each method of extraction works well in the laboratory, but analysts typically select their preferred method based on factors such as the cost, the time involved, the quantity of DNA yielded, and the quality of DNA yielded. 18. RFLP analysis. Restriction fragment length polymorphism. RFLP stands for restriction fragment length polymorphism and, in terms of DNA analysis, describes a DNA testing method which utilizes restriction enzymes to cut the DNA at short and specific sequences throughout the sample. To start off processing in the laboratory, the sample has to first go through an extraction protocol, which may vary depending on the sample type or laboratory SOPs, standard operating procedures. Once the DNA has been extracted from the cells within the sample and separated away from extraneous cellular materials and any nucleuses that would degrade the DNA, the sample can then be introduced to the desired restriction enzymes to be cut up into discernible fragments. Following the enzyme digestion, a Southern blot is performed. Southern blots are a size-based separation method that are performed on a gel with either radioactive or chemiluminescent probes. RFLP could be conducted with single-locus or multi-locus probes, probes which target either one location on the DNA or multiple locations on the DNA. Incorporating the multi-locus probes allowed for higher discrimination power for the analysis, however completion of this process could take several days to a week for one sample due to the extreme amount of time required by each step required for visualization of the probes. Polymerase chain reaction (PCR) analysis. This technique was developed in 1983 by Kary Mullis. PCR is now a common and important technique used in medical and biological research labs for a variety of applications. 19. PCR, or polymerase chain reaction, is a widely used molecular biology technique to amplify a specific DNA sequence. Steps of polymerase chain reaction. Amplification is achieved by a series of 3 steps. 1. Denaturation. In this step, the DNA is heated to 95 degrees Celsius to dissociate the hydrogen bonds between the complementary base pairs of the double-stranded DNA. 2. Annealing. During this stage the reaction is cooled to 50 to 65 degrees C. This enables the primers to attach to a specific location on the single-stranded template DNA by way of hydrogen bonding. 3. Extension. A thermostable DNA polymerase which is Taq polymerase is commonly used at this step. This is done at a temperature of 72 degrees Celsius. DNA polymerase adds nucleotides in the 5'-3" direction and synthesizes the complementary strand of the DNA template. STR analysis. Short tandem repeat STR analysis on a simplified model. First, a DNA sample undergoes polymerase chain reaction with primers targeting certain STRs, which vary in lengths between individuals and their alleles. The resultant fragments are separated by size, such as electrophoresis. 20. The system of DNA profiling used today is based on polymerase chain reaction (PCR) and uses simple sequences. 9. From country to country, different STR-based DNA profiling systems are in use. In North America, systems that amplify the CODES 20/21 core loci are almost universal, whereas in the United Kingdom the DNA 17 loci system is in use, and Australia uses 18 core markers. 22. The true power of STR analysis is in its statistical power of discrimination. Because the 20 loci that are currently used for discrimination in CODES are independently assorted, having a certain number of repeats at one locus does not change the likelihood of having any number of repeats at any other locus. The product rule for probabilities can be applied. This means that, if someone has the DNA type of ABC, where the 3 loci were independent, then the probability of that individual having that DNA type is the probability of having type A times the probability of having type B times the probability of having type C. This has resulted in the ability to generate match probabilities of 1 in a quintillion, 1x1018, or more. However, DNA database searches showed much more frequent than expected false DNA profile matches. 23. Y chromosome analysis. Due to the paternal inheritance, Y haplotypes provide information about the genetic ancestry of the male population. To investigate this population history, and to provide estimates for haplotype frequencies in criminal casework, the Y Haplotype Reference Database (YHRD) has been created in 2000 as an online resource. It currently comprises more than 300,000 minimal 8-locus haplotypes from worldwide populations. 24. Mitochondrial analysis. mtDNA can be obtained from such material as hair shafts and old bones teeth. 25. Control mechanism based on interaction point with data. This can be determined by tool placement in sample. 26. Issues with forensic DNA samples. When people think of DNA analysis, they often think about television shows like NCIS or CSI, which portray DNA samples coming into a lab and being instantly analyzed, followed by the pulling up of a picture of the suspect within minutes. However, the reality is quite different, and perfect DNA samples are often not collected from the scene of a crime. Homicide victims are frequently left exposed to harsh conditions before they are found, and objects that are used to commit crimes have often been handled by more than one person. The two most prevalent issues that forensic scientists encounter when analyzing DNA samples are degraded samples and DNA mixtures. 27. Degraded DNA. Before modern PCR methods existed, it was almost impossible to analyze degraded DNA samples. Methods like restriction fragment length polymorphism, RFLP, which was the first technique used for DNA analysis in forensic science, required high molecular weight DNA in the sample in order to get reliable data. High molecular weight DNA, however, is lacking in degraded samples, as the DNA is too fragmented to carry out RFLP accurately. It was only when polymerase chain reaction techniques were invented that analysis of degraded DNA samples were able to be carried out. Multiplex PCR in particular made it possible to isolate and to amplify the small fragments of DNA that are still left in degraded samples. When multiplex PCR methods are compared to the older methods like RFLP, a vast difference can be seen. Multiplex PCR can theoretically amplify less than 1 nanogram of DNA, but RFLP had to have at least 100 nanograms of DNA in order to carry out an analysis. 28. Low template DNA. Low template DNA can happen when there is less than 0.1 nanograms, 29, of DNA in a sample. This can lead to more stochastic effects, random events such as allelic dropout or allelic drop-in, which can alter the interpretation of a DNA profile. These stochastic effects can lead to the unequal amplification of the two alleles that come from a heterozygous individual. It is especially important to take low template DNA into account when dealing with a mixture of DNA sample. This is because for one or more of the contributors in the mixture, they are more likely to have less than the optimal amount of DNA for the PCR reaction to work properly. 30. Therefore, stochastic thresholds are developed for DNA profile interpretation. The stochastic threshold is the minimum peak height RFU value seen in an electropherogram where dropout occurs. If the peak height value is above this threshold, then it is reasonable to assume that allelic dropout has not occurred. For example, if only one peak is seen for a particular locus in the electropherogram but its peak height is above the stochastic threshold, then we can reasonably assume that this individual is homozygous and is not missing its heterozygous partner allele that otherwise would have dropped out due to having low template DNA. Allelic dropout can occur when there is low template DNA because there is such little DNA to start with that at this locus the contributor to the DNA sample, or mixture, is a true heterozygote but the other allele is not amplified and so it would be lost. Allelic drop-in 31 can also occur when there is low template DNA because sometimes the stutter peak can be amplified. The stutter is an artifact of PCR. During the PCR reaction, DNA polymerase will come in and add nucleotides off of the primer, but this whole process is very dynamic, meaning that the DNA polymerase is constantly binding, popping off and then rebinding. Therefore, sometimes DNA polymerase will rejoin at the short tandem repeat ahead of it, leading to a short tandem repeat that is one repeat less than the template. During PCR, if DNA polymerase happens to bind to a locus in stutter and starts to amplify it to make lots of copies, then this stutter product will appear randomly in the electropherogram, leading to allelic drop-in. Mini-STR analysis. In instances in which DNA samples are degraded, like if there are intense fires or all that remains are bone fragments, standard STR testing on those samples can be inadequate. When standard STR testing is done on highly degraded samples, the larger STR loci often drop out, and only partial DNA profiles are obtained. Partial DNA profiles can be a powerful tool, but the probability of a random match is larger than if a full profile was obtained. One method that has been developed to analyze degraded DNA samples is to use mini-STR technology. In the new approach, primers are specially designed to bind closer to the STR region. 32. In normal STR testing, the primers bind to longer sequences that contain the STR region within the segment. Mini-STR analysis, however, targets only the STR location, which results in a DNA product that is much smaller. 32. By placing the primers closer to the actual STR regions, there is a higher chance that successful amplification of this region will occur. Successful amplification of those STR regions can now occur, and more complete DNA profiles can be obtained. The success that smaller PCR products produce a higher success rate with highly degraded samples was first reported in 1995, when mini-STR technology was used to identify victims of the Waco fire. 33. DNA mixtures. Mixtures are another common issue faced by forensic scientists when they are analyzing unknown or questionable DNA samples. A mixture is defined as a DNA sample that contains two or more individual contributors. 28. That can often occur when a DNA sample is swabbed from an item that is handled by more than one person or when a sample contains both the victim's and the assailant's DNA. The presence of more than one individual in a DNA sample can make it challenging to detect individual profiles, and interpretation of mixtures should be performed only by highly trained individuals. Mixtures that contain 2 or 3 individuals can be interpreted with difficulty. Mixtures that contain 4 or more individuals are much too convoluted to get individual profiles. One common scenario in which a mixture is often obtained is in the case of sexual assault. A sample may be collected that contains material from the victim's consensual sexual partners, and the perpetrators. 34. Mixtures can generally be sorted into 3 categories: Type A, Type B, and Type C. 35. Type A mixtures have alleles with similar peak heights all around, so the contributors cannot be distinguished from each other. Type B mixtures can be deconvoluted by comparing peak height ratios to determine which alleles were donated together. Type C mixtures cannot be safely interpreted with current technology because the samples were affected by DNA degradation or having too small a quantity of DNA present. When looking at an electropherogram, it is possible to determine the number of contributors in less complex mixtures based on the number of peaks located in each locus. In comparison to a single source profile, which will only have 1 or 2 peaks at each locus, a mixture is when there are 3 or more peaks at 2 or more loci. 36. If there are 3 peaks at only a single locus, then it is possible to have a single contributor who is triallelic at that locus. 37. 2-person mixtures will have between 2 and 4 peaks at each locus, and 3-person mixtures will have between 3 and 6 peaks at each locus. Mixtures become increasingly difficult to deconvolute as the number of contributors increases. As detection methods in DNA profiling advance, forensic scientists are seeing more DNA samples that contain mixtures, as even the smallest contributor can now be detected by modern tests. The ease in which forensic scientists have in interpenetrating DNA mixtures largely depends on the ratio of DNA present from each individual, the genotype combinations, and the total amount of DNA amplified. 38. The DNA ratio is often the most important aspect to look at in determining whether a mixture can be interpreted. For example, if a DNA sample had 2 contributors, it would be easy to interpret individual profiles if the ratio of DNA contributed by one person was much higher than the second person. When a sample has 3 or more contributors, it becomes extremely difficult to determine individual profiles. Fortunately, advancements in probabilistic genotyping may make that sort of determination possible in the future. Probabilistic genotyping uses complex computer software to run through thousands of mathematical computations to produce statistical likelihoods of individual genotypes found in a mixture. 39. DNA profiling in plant. Plant DNA profiling fingerprinting is a method for identifying cultivars that uses molecular marker techniques. This method is gaining attention due to trade-related intellectual property rights (TRIPS) and the Convention on Biological Diversity (CBD). 40. Advantages of plant DNA profiling. Identification, authentication, specific distinction, detecting adulteration and identifying phytoconstituents are all possible with DNA fingerprinting in medical plants. 41. DNA-based markers are critical for these applications, determining the future of scientific study in pharmacognosy. 41. It also helps with determining the traits such as seed size and leaf color are likely to improve the offspring or not. 42. DNA databases. An early application of a DNA database was the compilation of a mitochondrial DNA concordance, 43, prepared by Kevin W. P. Miller and John L. Dawson at the University of Cambridge from 1996 to 1999, 44, from data collected as part of Miller's PhD thesis. There are now several DNA databases in existence around the world. Some are private, but most of the largest databases are government-controlled. The United States maintains the largest DNA database, with the Combined DNA Index System, CODIS, holding over 13 million records as of May 2018. 45. The United Kingdom maintains the National DNA Database, NDNAD, which is of similar size, despite the UK's smaller population. The size of this database, and its rate of growth, are giving concern to civil liberties groups in the UK, where police have wide-ranging powers to take samples and retain them even in the event of acquittal. 46. The Conservative Liberal Democrat coalition partially addressed these concerns with Part 1 of the Protection of Freedoms Act 2012, under which DNA samples must be deleted if suspects are acquitted or not charged, except in relation to certain, mostly serious or sexual, offenses. Public discourse around the introduction of advanced forensic techniques, such as genetic genealogy using public genealogy databases and DNA phenotyping approaches, has been limited, disjointed, unfocused, and raises issues of privacy and consent that may warrant the establishment of additional legal protections. 47. The U.S. Patriot Act of the United States provides a means for the U.S. government to get DNA samples from suspected terrorists. DNA information from crimes is collected and deposited into the CODIS database, which is maintained by the FBI. CODIS enables law enforcement officials to test DNA samples from crimes for matches within the database, providing a means of finding specific biological profiles associated with collected DNA evidence. 48. When a match is made from a national DNA databank to link a crime scene to an offender having provided a DNA sample to a database, that link is often referred to as a cold hit. A cold hit is of value in referring the police agency to a specific suspect but is of less evidential value than a DNA match made from outside the DNA databank. 49. FBI agents cannot legally store DNA of a person not convicted of a crime. DNA collected from a suspect not later convicted must be disposed of and not entered into the database. In 1998, a man residing in the UK was arrested on accusation of burglary. His DNA was taken and tested, and he was later released. 9 months later, this man's DNA was accidentally and illegally entered in the DNA database. New DNA is automatically compared to the DNA found at cold cases and, in this case, this man was found to be a match to DNA found at a rape and assault case 1 year earlier. The government then prosecuted him for these crimes. During the trial, the DNA match was requested to be removed from the evidence because it had been illegally entered into the database. The request was carried out. 50. The DNA of the perpetrator, collected from victims of rape, can be stored for years until a match is found. In 2014, to address this problem, Congress extended a bill that helps states deal with a backlog of evidence. 51. DNA profiling databases in plants. PIDS. PIDS, Plant International DNA Fingerprinting System, is an open-source web server and free software-based Plant International DNA fingerprinting system. It manages huge amount of microsatellite DNA fingerprint data, performs genetic studies, and automates collection, storage and maintenance while decreasing human error and increasing efficiency. The system may be tailored to specific laboratory needs, making it a valuable tool for plant breeders, forensic science, and human fingerprint recognition. It keeps track of experiments, standardizes data and promotes inter-database communication. It also helps with the regulation of variety quality, the preservation of variety rights and the use of molecular markers in breeding by providing location statistics, merging, comparison and genetic analysis function. 52. Considerations in evaluating DNA evidence. When using RFLP, the theoretical risk of a coincidental match is 1 in 100 billion, although the practical risk is actually 1 in 1,000 because monozygotic twins are 0.2% of the human population. 53. Moreover, the rate of laboratory error is almost certainly higher than that, and actual laboratory procedures often do not reflect the theory under which the coincidence probabilities were computed. For example, coincidence probabilities may be calculated based on the probabilities that markers in two samples have bands in precisely the same location, but a laboratory worker may conclude that similar but not precisely identical band patterns result from identical genetic samples with some imperfection in the agarose gel. However, in that case, the laboratory worker increases the coincidence risk by expanding the criteria for declaring a match. Studies conducted in the 2000s quoted relatively high error rates, which may be cause for concern. 54. In the early days of genetic fingerprinting, the necessary population data to compute a match probability accurately was sometimes unavailable. Between 1992 and 1996, arbitrary low ceilings were controversially put on match probabilities used in RFLP analysis, rather than the higher theoretically computed ones. 55. Evidence of genetic relationship. It is possible to use DNA profiling as evidence of genetic relationship, although such evidence varies in strength from weak to positive. Testing that shows no relationship is absolutely certain. Further, while almost all individuals have a single and distinct set of genes, ultra-rare individuals, known as chimeras, have at least two different sets of genes. There have been 2 cases of DNA profiling that falsely suggested that a mother was unrelated to her children. 56. Fake DNA evidence. The functional analysis of genes and their coding sequences, open reading frames (ORFs), typically requires that each ORF be expressed, the encoded protein purified, antibodies produced, phenotypes examined, intracellular localization determined, and interactions with other proteins sought. 57. In a study conducted by the life science company Nuclex and published in the journal Forensic Science International, scientists found that an in vitro synthesized sample of DNA matching any desired genetic profile can be constructed using standard molecular biology techniques without obtaining any actual tissue from that person. DNA evidence in criminal trials. Familial DNA searching. Familial DNA searching, sometimes referred to as familial DNA or familial DNA database searching, is the practice of creating new investigative leads in cases where DNA evidence found at the scene of a crime, forensic profile, strongly resembles that of an existing DNA profile, offender profile, in a state DNA database, but there is not an exact match. 58-59. After all other leads have been exhausted, investigators may use specially developed software to compare the forensic profile to all profiles taken from a state's DNA database to generate a list of those offenders already in the database who are most likely to be a very close relative of the individual whose DNA is in the forensic profile. 60. Familial DNA database searching was first used in an investigation leading to the conviction of Jeffrey Gaffor of the murder of Lynette White in the United Kingdom on the 4th of July 2003. DNA evidence was matched to Gaffor's nephew, who at 14 years old had not been born at the time of the murder in 1988. It was used again in 2004, 61, to find a man who threw a brick from a motorway bridge and hit a lorry driver, killing him. DNA found on the brick matched that found at the scene of a car theft earlier in the day, but there were no good matches on the national DNA database. A wider search found a partial match to an individual. On being questioned, this man revealed he had a brother, Craig Harmon, who lived very close to the original crime scene. Harmon voluntarily submitted a DNA sample, and confessed when it matched the sample from the brick. 62. As of 2011, familial DNA database searching is not conducted on a national level in the United States, where states determine how and when to conduct familial searches. The first familial DNA search with a subsequent conviction in the United States was conducted in Denver, Colorado, in 2008, using software developed under the leadership of Denver District Attorney Mitch Morrissey and Denver Police Department Crime Lab Director Greg Lauberg. 63. California was the first state to implement a policy for familial searching under then Attorney General Jerry Brown, who later became governor. 64. In his role as consultant to the Familial Search Working Group of the California Department of Justice, former Alameda County Prosecutor Rock Harmon is widely considered to have been the catalyst in the adoption of familial search technology in California. The technique was used to catch the Los Angeles serial killer known as the Grim Sleeper in 2010. 65. It was not a witness or informant that tipped off law enforcement to the identity of the Grim Sleeper serial killer, who had eluded police for more than 2 decades, but DNA from the suspect's own son. The suspect's son had been arrested and convicted in a felony weapons charge and swabbed for DNA the year before. When his DNA was entered into the database of convicted felons, detectives were alerted to a partial match to evidence found at the Grim Sleeper crime scenes. David Franklin Jr., also known as the Grim Sleeper, was charged with 10 counts of murder and 1 count of attempted murder. 66. More recently, familial DNA led to the arrest of 21-year-old Elvis Garcia on charges of sexual assault and false imprisonment of a woman in Santa Cruz in 2008. 67. In March 2011, Virginia Governor Bob McDonnell announced that Virginia would begin using familial DNA searches. 68. At a press conference in Virginia on the 7th of March 2011, regarding the East Coast rapist, Prince William County Prosecutor Paul Ebert and Fairfax County Police Detective John Kelly said the case would have been solved years ago if Virginia had used familial DNA searching. Aaron Thomas, the suspected East Coast rapist, was arrested in connection with the rape of 17 women from Virginia to Rhode Island, but familial DNA was not used in the case. 69. Critics of familial DNA database searches argue that the technique is an invasion of an individual's Fourth Amendment rights. 70. Privacy advocates are petitioning for DNA database restrictions, arguing that the only fair way to search for possible DNA matches to relatives of offenders or arrestees would be to have a population-wide DNA database. 50. Some scholars have pointed out that the privacy concerns surrounding familial searching are similar in some respects to other police search techniques. 71. And most have concluded that the practice is constitutional. 72. The Ninth Circuit Court of Appeals in United States v. Poole, vacated as moot, suggested that this practice is somewhat analogous to a witness looking at a photograph of one person and stating that it looked like the perpetrator, which leads law enforcement to show the witness photos of similar-looking individuals, one of whom was identified as the perpetrator. 73. Critics also state that racial profiling could occur on account of familial DNA testing. In the United States, the conviction rates of racial minorities are much higher than that of the overall population. It is unclear whether this is due to discrimination from police officers in the courts, as opposed to a simple higher rate of offense among minorities. Arrest-based databases, which are found in the majority of the United States, lead to an even greater level of racial discrimination. An arrest, as opposed to conviction, relies much more heavily on police discretion. 50. For instance, investigators with Denver District Attorney's Office successfully identified a suspect in a property theft case using a familial DNA search. In this example, the suspect's blood left at the scene of the crime strongly resembled that of a current Colorado Department of Corrections prisoner. 63. Partial matches. Partial DNA matches are the result of moderate stringency CODIS searches that produce a potential match that shares at least one allele at every locus. 74. Partial matching does not involve the use of familial search software, such as those used in the United Kingdom and the United States, or additional YSTR analysis and therefore often misses sibling relationships. Partial matching has been used to identify suspects in several cases in both countries. 75. And has also been used as a tool to exonerate the falsely accused. Darrell Hunt was wrongly convicted in connection with the rape and the murder of a young woman in 1984 in North Carolina. 76. Surreptitious DNA collecting. Police forces may collect DNA samples without a suspect's knowledge, and use it as evidence. The legality of the practice has been questioned in Australia. 77. In the United States, where it has been accepted, courts often rule that there is no expectation of privacy and cite California v. Greenwood, 1988, in which the Supreme Court held that the Fourth Amendment does not prohibit the warrantless search and seizure of garbage left for collection outside the curtilage of a home. Critics of this practice underline that this analogy ignores that most people have no idea that they risk surrendering their genetic identity to the police by, for instance, failing to destroy a used coffee cup. Moreover, even if they do realize it, there is no way to avoid abandoning one's DNA in public. 78. The United States Supreme Court ruled in Maryland v. King 2013 that DNA sampling of prisoners arrested for serious crimes is constitutional. 79. 80. 81. In the United Kingdom, the Human Tissue Act 2004 prohibits private individuals from covertly collecting biological samples, hair, fingernails, etc. for DNA analysis but exempts medical and criminal investigations from the prohibition. 82. England and Wales. Evidence from an expert who has compared DNA samples must be accompanied by evidence as to the sources of the samples and the procedures for obtaining the DNA profiles. 83. The judge must ensure that the jury must understand the significance of DNA matches and mismatches in the profiles. The judge must also ensure that the jury does not confuse the match probability, the probability that a person that is chosen at random has a matching DNA profile to the sample from the scene, with the probability that a person with matching DNA committed the crime. In 1996 R. vs. Doheny, 84. Juries should weigh up conflicting and corroborative evidence, using their own common sense and not by using mathematical formulae, such as Bayes' theorem, so as to avoid confusion, misunderstanding and misjudgment. 85. Presentation and evaluation of evidence of partial or incomplete DNA profiles. In R. vs. Bates, 86, Morbic L.J. said, "We can see no reason why partial profile DNA evidence should not be admissible provided that the jury are made aware of its inherent limitations and are given a sufficient explanation to enable them to evaluate it. There may be cases where the match probability in relation to all the samples tested is so great that the judge would consider its probative value to be minimal and decide to exclude the evidence in the exercise of his discretion, but this gives rise to no new question of principle and can be left for decision on a case-by-case basis. However, the fact that there exists in the case of all partial profile evidence the possibility that a missing allele might exculpate the accused altogether does not provide sufficient grounds for rejecting such evidence. In many there is a possibility, at least in theory, that evidence that would assist the accused and perhaps even exculpate him altogether exists, but that does not provide grounds for excluding relevant evidence that is available and otherwise admissible, though it does make it important to ensure that the jury are given sufficient information to enable them to evaluate that evidence properly. 87. DNA testing in the United States. CBP chemist reads a DNA profile to determine the origin of a commodity. There are state laws on DNA profiling in all 50 states of the United States. 88. Detailed information on database laws in each state can be found at the National Conference of State Legislatures website. 89. 2009. Scientists in Israel raised serious doubts concerning the use of DNA by law enforcement as the ultimate method of identification. In a paper published in the journal Forensic Science International, Genetics, the Israeli researchers demonstrated that it is possible to manufacture DNA in a laboratory, thus falsifying DNA evidence. The scientists fabricated saliva and blood samples, which originally contained DNA from a person other than the supposed donor of the blood and saliva. 90. The researchers also showed that, using a DNA database, it is possible to take information from a profile and manufacture DNA to match it, and that this can be done without access to any actual DNA from the person whose DNA they are duplicating. The synthetic DNA oligos required for the procedure are common in molecular laboratories. 90. The New York Times quoted the lead author, Daniel Frumkin, saying, "You can just engineer a crime scene. Any biology undergraduate could perform this." 90. Frumkin perfected a test that can differentiate real DNA samples from fake ones. His test detects epigenetic modifications, in particular, DNA methylation. 91. 70% of the DNA in any human genome is methylated, meaning it contains methyl group modifications within a CpG dinucleotide context. Methylation at the promoter region is associated with gene silencing. The synthetic DNA lacks this epigenetic modification, which allows the test to distinguish manufactured DNA from genuine DNA. 90. It is unknown how many police departments, if any, currently use the test. No police lab has publicly announced that it is using the new test to verify DNA results. 92. Researchers at the University of Tokyo integrated an artificial DNA replication scheme with a rebuilt gene expression system and microcompartmentalization utilizing cell-free materials alone for the first time. Multiple cycles of serial dilution were performed on a system contained in microscale water and oil droplets. 93. Chances of making DNA change on purpose. Overall, this study's artificial genomic DNA, which kept copying itself using self-encoded proteins and made its sequence better on its own, is a good starting point for making more complex artificial cells. By adding the genes needed for transcription and translation to artificial genomic DNA, it may be possible in the future to make artificial cells that can grow on their own when fed small molecules like amino acids and nucleotides. Using living organisms to make useful things, like drugs and food, would be more stable and easier to control in these artificial cells. 93. On July 7, 2008, the American Chemical Society reported that Japanese chemists have created the world's first DNA molecule comprised nearly completely of synthetic components. A nanoparticle-based artificial transcription factor for gene regulation. NanoScript is a nanoparticle-based artificial transcription factor that is supposed to replicate the structure and function of TFs. On gold nanoparticles, functional peptides and tiny molecules referred to as synthetic transcription factors, which imitate the various TF domains, were attached to create NanoScript. We show that NanoScript localizes to the nucleus and begins transcription of a reporter plasmid by an amount more than 15-fold. Moreover, NanoScript can successfully transcribe targeted genes onto endogenous DNA in a non-viral manner. 94. 3 different fluorophores, red, green, and blue, were carefully fixed on the DNA rod surface to provide spatial information and create a nanoscale barcode. Epifluorescence and total internal reflection fluorescence microscopy reliably deciphered spatial information between fluorophores. By moving the 3 fluorophores on the DNA rod, this nanoscale barcode created 216 fluorescence patterns. 95. Cases. In 1986, Richard Buckland was exonerated, despite having admitted to the rape and murder of a teenager near Leicester, the city where DNA profiling was first developed. This was the first use of DNA fingerprinting in a criminal investigation, and the first to prove a suspect's innocence. 96. The following year Colin Pitchfork was identified as the perpetrator of the same murder, in addition to another, using the same techniques that had cleared Buckland. 97. In 1987, genetic fingerprinting was used in a U.S. criminal court for the first time in the trial of a man accused of unlawful intercourse with a mentally disabled 14-year-old female who gave birth to a baby. 98. In 1987, Florida rapist Tommy Lee Andrews was the first person in the United States to be convicted as a result of DNA evidence for raping a woman during a burglary. He was convicted on the 6th of November 1987 and sentenced to 22 years in prison. 99. 100. In 1990, a violent murder of a young student in Brno was the first criminal case in Czechoslovakia solved by DNA evidence, with the murderer sentenced to 23 years in prison. 101. 102. In 1992, DNA from a Palo Verde tree was used to convict Mark Allen Bogan of murder. DNA from seed pods of a tree at the crime scene was found to match that of seed pods found in Bogan's truck. This is the first instance of plant DNA admitted in a criminal case. 103. 104. 105. In 1994, the claim that Anna Anderson was Grand Duchess Anastasia Nikolaevna of Russia was tested after her death using samples of her tissue that had been stored at a Charlottesville hospital following a medical procedure. The tissue was tested using DNA fingerprinting, and showed that she bore no relation to the Romanovs. 106. In 1994, Earl Washington Jr. of Virginia had his death sentence commuted to life imprisonment a week before his scheduled execution date based on DNA evidence. He received a full pardon in 2000 based on more advanced testing. 107. In 1999, Raymond Easton, a disabled man from Swindon, England, was arrested and detained for 7 hours in connection with a burglary. He was released due to an inaccurate DNA match. His DNA had been retained on file after an unrelated domestic incident some time previously. 108. In 2000, Frank Lee Smith was proved innocent by DNA profiling of the murder of an 8-year-old girl after spending 14 years on death row in Florida, USA. However, he had died of cancer just before his innocence was proven. 109. In view of this, the Florida state governor ordered that in future any death row inmate claiming innocence should have DNA testing. 107. In May 2000, Gordon Graham murdered Paul Galt at his home in Lisburn, Northern Ireland. Graham was convicted of the murder when his DNA was found on a sports bag left in the house as part of an elaborate ploy to suggest the murder occurred after a burglary had gone wrong. Graham was having an affair with the victim's wife at the time of the murder. It was the first time low copy number DNA was used in Northern Ireland. 110. In 2001, Wayne Butler was convicted for the murder of Celia Doughty. It was the first murder in Australia to be solved using DNA profiling. 111-112. In 2002, the body of James Hanratty, hanged in 1962 for the A6 murder, was exhumed and DNA samples from the body and members of his family were analyzed. The results convinced Court of Appeal judges that Hanratty's guilt, which had been strenuously disputed by campaigners, was proved beyond doubt. 113 Paul Foot and some other campaigners continued to believe in Hanratty's innocence and argued that the DNA evidence could have been contaminated, noting that the small DNA samples from items of clothing, kept in a police laboratory for over 40 years, in conditions that do not satisfy modern evidential standards, had to be subjected to very new amplification techniques in order to yield any genetic profile. 114 However, no DNA other than Hanratty's was found on the evidence tested, contrary to what would have been expected had the evidence indeed been contaminated. 115. In August 2002, Annalisa Vicentini was shot dead in Tuscany. Bartender Peter Hampkin, 23, was arrested in Merseyside in March 2003 on an extradition warrant heard at Bow Street Magistrates Court in London to establish whether he should be taken to Italy to face a murder charge. DNA proved he shot her, but he was cleared on other evidence. 116. In 2003, Welshman Geoffrey Gafford was convicted of the 1988 murder of Lynette White, when crime scene evidence collected 12 years earlier was re-examined using STR techniques, resulting in a match with his nephew. 117. In June 2003, because of new DNA evidence, Dennis Halstead, John Kogut and John Restivo won a retrial on their murder conviction. Their convictions were struck down and they were released. 118. In 2004, DNA testing shed new light into the mysterious 1912 disappearance of Bobby Dunbar, a 4-year-old boy who vanished during a fishing trip. He was allegedly found alive 8 months later in the custody of William Cantwell Walters, but another woman claimed that the boy was her son, Bruce Anderson, whom she had entrusted in Walters' custody. The courts disbelieved her claim and convicted Walters for the kidnapping. The boy was raised and known as Bobby Dunbar throughout the rest of his life. However, DNA tests on Dunbar's son and nephew revealed the two were not related, thus establishing that the boy found in 1912 was not Bobby Dunbar, whose real fate remains unknown. 119. 2005, Gary Leiterman was convicted of the 1969 murder of Jane Mixer, a law student at the University of Michigan, after DNA found on Mixer's pantyhose was matched to Leiterman. DNA in a drop of blood on Mixer's hand was matched to John Rulas, who was only 4 years old in 1969 and was never successfully connected to the case in any other way. Leiterman's defense unsuccessfully argued that the unexplained match of the blood spot to Rulas pointed to cross-contamination and raised doubts about the reliability of the lab's identification of Leiterman. 120-121. In November 2008, Anthony Curcio was arrested for masterminding one of the most elaborately planned armored car heists in history. DNA evidence linked Curcio to the crime. 122. In March 2009, Sean Hodgson, convicted of 1979 killing of Teresa DeSimone, 22, in her car in Southampton, was released after tests proved DNA from the scene was not his. It was later matched to DNA retrieved from the exhumed body of David Lace. Lace had previously confessed to the crime but was not believed by the detectives. He served time in prison for other crimes committed at the same time as the murder and then committed suicide in 1988. 123. In 2012, a case of babies being switched, many decades earlier, was discovered by accident. After undertaking DNA testing for other purposes, Alice Collins Plabuch was advised that her ancestry appeared to include a significant Ashkenazi Jewish component, despite a belief in her family that they were of predominantly Irish descent. Profiling of Plabuch's genome suggested that it included distinct and unexpected components associated with Ashkenazi, Middle Eastern, and Eastern European populations. This led Plabuch to conduct an extensive investigation, after which she concluded that her father had been switched, possibly accidentally, with another baby soon after birth. Plabuch was also able to identify the biological ancestors of her father. 124-125. In 2016, Anthea Ring, abandoned as a baby, was able to use a DNA sample and DNA matching database to discover her deceased mother's identity and roots in County Mayo, Ireland. A recently developed forensic test was subsequently used to capture DNA from saliva left on old stamps and envelopes by her suspected father, uncovered through painstaking genealogy research. The DNA in the first 3 samples was too degraded to use. However, on the 4th, more than enough DNA was found. The test, which has a degree of accuracy acceptable in UK courts, proved that a man named Patrick Coyne was her biological father. 126-127. In 2018, the Buckskin Girl, a body found in 1981 in Ohio, was identified as Marcia King from Arkansas using DNA genealogical techniques. 128. In 2018, Joseph James D'Angelo was arrested as the main suspect for the Golden State Killer using DNA and genealogy techniques. 129. In 2018, William Earl Talbot II was arrested as a suspect for the 1987 murders of Jay Cook and Tanya Van Cuylenberg with the assistance of genealogical DNA testing. The same genetic genealogist that helped in this case also helped police with 18 other arrests in 2018. 130. In 2018, with the use of next-generation identification systems' enhanced biometric capabilities, the FBI matched the fingerprint of a suspect named Timothy David Nelson and arrested him 20 years after the alleged sexual assault. 131. DNA evidence is evidence to prove rights of succession to British titles. DNA testing has been used to establish the right of succession to British titles. 132 cases. Baron Moynihan. Pringle baronets. DNA barcoding. DNA barcoding is a method of species identification using a short section of DNA from a specific gene or genes. The premise of DNA barcoding is that by comparison with a reference library of such DNA sections, also called sequences, an individual sequence can be used to uniquely identify an organism to species, just as a supermarket scanner uses the familiar black stripes of the UPC barcode to identify an item in its stock against its reference database. 1. These barcodes are sometimes used in an effort to identify unknown species or parts of an organism, simply to catalog as many taxa as possible, or to compare with traditional taxonomy in an effort to determine species boundaries. 2. DNA barcoding scheme. Different gene regions are used to identify the different organismal groups using barcoding. The most commonly used barcode region for animals and some protists is a portion of the cytochrome C oxidase COI or COX-1 gene, found in mitochondrial DNA. Other genes suitable for DNA barcoding are the internal transcribed spacer, its rRNA often used for fungi and rubisco used for plants. 3, 4, 5 microorganisms are detected using different gene regions. The 16S rRNA gene, for example, is widely used in identification of prokaryotes, whereas the 18S rRNA gene is mostly used for detecting microbial eukaryotes. These gene regions are chosen because they have less interspecific within species variation than interspecific between species variation, which is known as a barcoding gap. 6. Some applications of DNA barcoding include identifying plant leaves even when flowers or fruits are not available, identifying pollen collected on the bodies of pollinating animals, identifying insect larvae which may have fewer diagnostic characters than adults, or investigating the diet of an animal based on its stomach content, saliva or feces. 7. When barcoding is used to identify organisms from a sample containing DNA from more than one organism, the term DNA metabarcoding is used. 8, 9, e.g. DNA metabarcoding of diatom communities in rivers and streams, which is used to assess water quality. 10. Background. DNA barcoding techniques were developed from early DNA sequencing work on microbial communities using the 5S rRNA gene. 11. In 2003, specific methods and terminology of modern DNA barcoding were proposed as a standardized method for identifying species, as well as potentially allocating unknown sequences to higher taxa such as orders and phyla, in a paper by Paul D. and Hebert et al., from the University of Guelph, Ontario, Canada. 12. Hebert and his colleagues demonstrated the utility of the cytochrome C oxidase COI gene, first utilized by Fulmer et al. In 1994, using their published DNA primers as a tool for phylogenetic analyses at the species levels. 12. As a suitable discriminatory tool between metazoa and invertebrates. 13. The Fulmer region of the COI gene is commonly used for distinction between taxa based on its patterns of variation at the DNA level. The relative ease of retrieving the sequence, and variability mixed with conservation between species, are some of the benefits of COI. Calling the profiles, barcodes, Hebert et al. Envisaged the development of a COI database that could serve as a basis for a global bioidentification system. Methods. Sampling and preservation. Barcoding can be done from tissue from a target specimen, from a mixture of organisms bulk sample, or from DNA present in environmental samples, e.g. water or soil. The methods for sampling, preservation or analysis differ between those different types of sample. Tissue samples. To barcode a tissue sample from the target specimen, a small piece of skin, a scale, a leg or antenna is likely to be sufficient depending on the size of the specimen. To avoid contamination, it is necessary to sterilize used tools between samples. It is recommended to collect 2 samples from one specimen, one to archive, and one for the barcoding process. Sample preservation is crucial to overcome the issue of DNA degradation. Bulk samples. A bulk sample is a type of environmental sample containing several organisms from the taxonomic group under study. The difference between bulk samples, in the sense used here, and other environmental samples is that the bulk sample usually provides a large quantity of good quality DNA. Examples of bulk samples include aquatic macroinvertebrate samples collected by KickNet, or insect samples collected with a Malaise trap. Filtered or size fractionated water samples containing whole organisms like unicellular eukaryotes are also sometimes defined as bulk samples. Such samples can be collected by the same techniques used to obtain traditional samples for morphology-based identification. EDNA samples. The environmental DNA EDNA method is a non-invasive approach to detect and identify species from cellular debris or extracellular DNA present in environmental samples, e.g. water or soil, through barcoding or metabarcoding. The approach is based on the fact that every living organism leaves DNA in the environment, and this environmental DNA can be detected even for organisms that are at very low abundance. Thus, for field sampling, the most crucial part is to use DNA-free material and tools on each sampling site or sample to avoid contamination, if the DNA of the target organisms is likely to be present in low quantities. On the other hand, an EDNA sample always includes the DNA of whole cell, living microorganisms, which are often present in large quantities. Therefore, microorganism samples taken in the natural environment also are called EDNA samples, but contamination is less problematic in this context due to the large quantity of target organisms. The EDNA method is applied on most sample types, like water, sediment, soil, animal feces, stomach content or blood from e.g. leeches. 14. DNA extraction, amplification and sequencing. DNA barcoding requires that DNA in the sample is extracted. Several different DNA extraction methods exist, and factors like cost, time, sample type and yield affect the selection of the optimal method. When DNA from organismal or EDNA samples is amplified using polymerase chain reaction (PCR), the reaction can be affected negatively by inhibitor molecules contained in the sample. 15. Removal of these inhibitors is crucial to ensure that high-quality DNA is available for subsequent analyzing. Amplification of the extracted DNA is a required step in DNA barcoding. Typically, only a small fragment of the total DNA material is sequenced, typically 400 to 800 base pairs, 16, to obtain the DNA barcode. Amplification of EDNA material is usually focused on smaller fragment sizes, less than 200 base pairs, as EDNA is more likely to be fragmented than DNA material from other sources. However, some studies argue that there is no relationship between amplicon size and detection rate. Of EDNA, 1718. HiSeq sequencers at SciLife Lab in Uppsala, Sweden. The photo was taken during the excursion of SLU course PNS 0169 in March 2019. When the DNA barcode marker region has been amplified, the next step is to sequence the marker region using DNA sequencing methods. 19. Many different sequencing platforms are available, and technical development is proceeding rapidly. Marker selection. A schematic view of primers in target region, demonstrated on 16S-rRNA gene in Pseudomonas. As primers, one typically selects short conserved sequences with low variability, which can thus amplify most or all species in the chosen target group. The primers are used to amplify a highly variable target region in between the two primers, which is then used for species discrimination. Modified from variable copy number, intragenomic heterogeneities and lateral transfers of the 16S-rRNA gene in Pseudomonas by Botelis, Jocelyn, Enseed Milo, Sandrine, Bessori, Ludovic, Quillet, Laurent, used under CC BY, available from rRNA gene in. Markers used for DNA barcoding are called barcodes. In order to successfully characterize species based on DNA barcodes, selection of informative DNA regions is crucial. A good DNA barcode should have low intraspecific and high interspecific variability, 12, and possess conserved flanking sites for developing universal PCR primers for wide taxonomic application. The goal is to design primers that will detect and distinguish most or all the species in the studied group of organisms, high taxonomic resolution. The length of the barcode sequence should be short enough to be used with current sampling source, DNA extraction, amplification and sequencing methods. 20. Ideally, one gene sequence would be used for all taxonomic groups, from viruses to plants and animals. However, no such gene region has been found yet, so different barcodes are used for different groups of organisms, or depending on the study question. For animals, the most widely used barcode is mitochondrial cytochrome C oxidase I, COI, locus. 21. Other mitochondrial genes, such as CYTB, 12S or 16S, are also used. Mitochondrial genes are preferred over nuclear genes because of their lack of introns, their haploid mode of inheritance and their limited recombination. 21. 22. Moreover, each cell has various mitochondria, up to several thousand, and each of them contains several circular DNA molecules. Mitochondria can therefore offer abundant source of DNA even when sample tissue is limited. In plants, however, mitochondrial genes are not appropriate for DNA barcoding because they exhibit low mutation rates. 23. A few candidate genes have been found in the chloroplast genome, the most promising being maturase K gene, MATK, by itself or in association with other genes. Multi-locus markers such as ribosomal internal transcribed spacers, its DNA, along with MATK, RBCL, tRNH or other genes have also been used for species identification. The best discrimination between plant species has been achieved when using 2 or more chloroplast barcodes. 24. For bacteria, the small subunit of ribosomal RNA, 16S gene can be used for different taxa, as it is highly conserved. 25. Some studies suggest COI, 26 type 2 chaperonin, CPN60, 27, or beta subunit of RNA polymerase, RPOB, 28, also could serve as bacterial DNA barcodes. Barcoding fungi is more challenging, and more than one primer combination might be required. 29. The COI marker performs well in certain fungi groups, 30, but not equally well in others. 31. Therefore, additional markers are being used, such as its rDNA and the large subunit of nuclear ribosomal RNA, 28 SLSURRNA. 32. Within the group of protists, various barcodes have been proposed, such as the D1-D2 or D2-D3 regions of 28S rDNA, V4 subregion of 18S rRNA gene, its rDNA and COI. Additionally, some specific barcodes can be used for photosynthetic protists, for example the large subunit of ribulose 1,5-bisphosphate carboxylase oxygenase gene, RBCL, and the chloroplastic 23S rRNA gene. Reference libraries and bioinformatics. Reference libraries are used for the taxonomic identification, also called annotation, of sequences obtained from barcoding or metabarcoding. These databases contain the DNA barcodes assigned to previously identified taxa. Most reference libraries do not cover all species within an organism group, and new entries are continually created. In the case of macro and many microorganisms, such as algae, these reference libraries require detailed documentation, sampling location and date, person who collected it, image, etc. And authoritative taxonomic identification of the voucher specimen, as well as submission of sequences in a particular format. However, such standards are fulfilled for only a small number of species. The process also requires the storage of voucher specimens in museum collections, herbaria and other collaborating institutions. Both taxonomically comprehensive coverage and content quality are important for identification accuracy. 33. In the microbial world, there is no DNA information for most species names, and many DNA sequences cannot be assigned to any Linnaean binomial. 34. Several reference databases exist depending on the organism group and the genetic marker used. There are smaller, national databases, e.g., FINBOL, and large consortia like the International Barcode of Life Project, IBOL. 35. Bold. Launched in 2007, the Barcode of Life data system BOLD 36 is one of the biggest databases, containing about 780,000 BINs barcode index numbers in 2022. It is a freely accessible repository for the specimen and sequence records for barcode studies, and it is also a workbench aiding the management, quality assurance and analysis of barcode data. The database mainly contains bin records for animals based on the COI genetic marker. For plant identification, BOLD accepts sequences from MATK and RBCL. Unite.